Clemson University professor James Morris received a $184,000 grant from the National Institutes of Health to research a method of starving the deadly parasites that threaten millions of people worldwide. If successful, the research could lead to the development of treatments for African sleeping sickness, a lethal infection caused by a parasite passed to humans from the bite of a tsetse fly. Current treatments are often unavailable, difficult to administer, or even deadly to patients. Morris hopes to develop affordable oral medications that can cure the disease with one dose.
If we have destabilized central governments in Central Africa and Sub-Saharan Africa, you have more of a problem. So as civil wars die down, things tend to get better because we're able to kill the fly or control the fly, and that controls the transmission and treat people. But last year, I think there were less than 10,000 human cases, which is a great year. But there have been outbreaks in modern times that have involved hundreds of thousands of people, and without a treatment, it's a lethal infection. Sleeping sickness is a problem in Sub-Saharan Africa, which is true. I think we have to be a little careful, though, because the American trypanosome, which causes Chagas disease, is in the U.S. now, and it's a bit of a different disease, but similar, kind of a cousin to an African trypanosome, and the drugs for it are terrible, too. We think we'll be able to apply what we find in our study to probably the American trypanosome, but more likely Leishmania, which is another cousin that causes something called Baghdad boil, which our troops have come back from Afghanistan and Iraq with. It's this sort of ulcerative wound that doesn't heal, that requires generally surgery to fix. And those guys are infected with the parasite. And so we do think we're going to have opportunities to treat a couple of related parasites because they all have the same general funny requirement for food.
These trypanosome parasites that cause African sleeping sickness are members of the class Kinetoplastia, which caused an estimated 1.4 million cases of disease in 2015, according to the World Health Organization. Morris will lead a multidisciplinary team of scientists from Brigham Young University, University of Wisconsin, Ohio State University, and the University of Kansas to evaluate around 25,000 different molecules that may be able to cut off the parasites from their only source of energy, sugar.
We started developing tools to measure sugar in live parasites, and that we had a great deal of fun doing it, and we made some real progress. And now using a combination of microscopy and other techniques that involve, fluorescence and these sorts of tools, we can measure what the parasite does when you add glucose to it or take it away in live time and see the cell respond. And we've learned a lot about biology that way. But, in the back of my mind, I kept thinking, gosh, we have a tool now to also, if we interfere with that biology, we could measure that too, right? If we block the ability of the parasite to take up its food, glucose, the sensor shouldn't work right, and we should be able to see that by either microscopy or some other technique and find small molecules that block it. You plug the hole and glucose can't get in, and they're going to die of starvation.
Scientists have conducted the genomic sequencing to identify the proteins parasites use to consume glucose from their hosts. What they don't know, yet, is how that glucose gets to the small organelles or sacs called glycosomes that parasites use to metabolize the sugar.
We don't have glycosomes, these little sacs. So, ta-da, you have a drug target. So we're going to look for things that inhibit both glucose uptake into the cell through that protein and then glucose uptake into the sac. And we'll get both. And that's, a lot of folks have worked on the one that takes stuff up from the outside into the cell. And that's been, folks have said we should make a drug to that for many, many years. And success has been, mixed. Okay, so I think it's still a reasonable thing to find new drugs for that, but in that we don't know how it gets into the sac at all, and that structure is so different than what we have. I think the likelihood of some success there is really higher. The parasite is entirely reliant on glucose, on the sugar. That's all it uses. So most cells, like us, use sugar and can eat other foods, fatty acids and amino acids and all kinds of stuff, but not the trypanosome when it's in your blood. And for decades, people have looked at that and said, well, that really is an Achilles heel. But the question has been, how do you attack it? And so maybe this is a new way, and we'll see what happens.